Okay, so it's time to move to the next speaker, and the next speaker is Crina Nimigean, and the title of the talk is Ball and Chain Inactivation in Potassium Channels. So please, Crina.
Hello, hi everybody. I guess I can share my screen now. Hopefully this is shared okay. Good morning, everybody, or actually good afternoon if you're in Rome, which is where I was hoping to be right now, but you know, alas. So I would like to first thank Alberto and Luigi and the organizers of this meeting for inviting me to speak, and I would like to thank Grazia for introducing me. And what I will talk to you about today is a story of ball and chain inactivation that we discovered in potassium channels as we were studying calcium gating of a particular channel, MTHK, calcium activated potassium channel. So first, a little bit about inactivation. What is inactivation? So one of the most famous inactivation in physiology is the sodium channel inactivation that happens during the generation of an action potential. So during the depolarization phase, you can see hopefully you can see my pointer. During the depolarization phase, sodium channels open and then immediately inactivate, allowing the membrane to repolarize during an action potential. So basically what inactivation is the stopping or the closure of the ion conduction pathway during the time that the stimulus is still applied. For instance, to use again the membrane depolarization as a stimulus, when one depolarizes the membrane from -70 to 0 millivolts during the depolarizing pulse, what you will see if you measure sodium channel current, you will see a fast activation and then a rapid inactivation during the time that the depolarization is still applied. Now, in potassium channels, there's also an N-type or ball and chain inactivation, and that also has an important physiological role in electrical signaling, in repetitive firing, to actually shape the action potential in such a way that during a train of action potentials in repetitive firing, the 100th action potential is actually much broader than that of the first one. This mechanism of N-type or ball and chain activation was dissected in the early 90s by Aldrich's lab, where they basically looked at deletion mutants in deletion constructs of shaker potassium channels, the same channels that you've heard about from Pancho in the previous talk. So basically, shaker potassium channels, upon application of a depolarizing pulse, and this is a figure from that paper, Hoshi et al. In 1990, you can see that they open quickly and then they no longer open, right? So this is a collection of single channel currents from different constructs, okay? The first one is the shaker potassium channel wild type. If you delete any of the first 20 constructs where any of the first 20 amino acids are deleted, what you can see that upon depolarization pulses, the channels are constantly open. They no longer inactivate like in the first 5 traces. And they propose a mechanism at that time that is shown on the bottom here, where the N-terminal 20 amino acids or so form a so-called ball, which is shown here as a sphere, as a yellow sphere, that is attached to the tetramer, and this is a view of the potassium channel from the bottom, and this is shown at rest, so basically the conduction is closed. When the channel opens upon some stimulus, in this case voltage obviously, one of these balls binds to its receptor site, which at that point was not known what the receptor site was, whether it was the pore, the gate, the mouth of the pore, some other place in the channel that would allosterically lead to channel closure and block ion conduction. Okay, the latest in conclusion, after many years of studying of N-type or ball and chain inactivation in potassium channels, the most recent findings were the ones that from McKinnon and Zhu and McKinnon in 2001, which were commented on by Rick Aldrich also in 2001, where basically they proposed, based on a mutant cycle analysis and blocker competition analysis in a shaker potassium channel that inactivates due to the inactivation domain of a beta subunit, not of its own N-terminus, but the N-terminus of an accessory beta subunit, shown here in blue. The ball and chain was proposed to actually bind and block in the pore of sterically blocking the pore of this potassium channel. But there was no structural correlate of ball and chain inactivation to date, meaning that there was no such structure of a channel in a ball and chain inactivated state. So this is where we come in. And what we were doing, and we were doing this for a while now, we're trying to figure out where the location of the gates, of the calcium gates, where the channel responds to calcium in large conductance calcium activated potassium channels. And there are two members of this family, of the large conductance calcium activated K channel family. One of them is the MTHK, which is a prokaryotic channel whose structure was determined way long time ago in 2002 in the McKinnon lab. And basically the structure was that of an open state calcium bound channel. It's a mutant where the second initiation site is actually mutated to actually form a simple. Basically, the idea in MTHK channels, we have a full-length channel that is formed, that forms the pore and one of the RCK domains here in light green, and then the second translation site, internal translation site in the gene, forms the soluble correspondent of this RCK domain, so the two of them interact together, forming a dimer, and that assembles within the final tetramer as an octameric gating ring. So this is an octameric gating ring that actually binds calcium, and this is the pore that this octameric gating ring gates when calcium is bound. So the intracellular gate of this channel, of the MTHK channel, the presence of calcium, is wide open. Okay, so it's open for passage of potassium, and there is no closed state. Now, the second member of the family is the BK channel, is a eukaryotic BK channel, which has, in addition to an RCK gating ring that is homologous to that of the MTHK, and in addition to the pore domain, which is also homologous to that of MTHK, they also have voltage sensor domains that you have heard in the previous talk. So the voltage sensing domains also modulate the opening of this pore. So both calcium binding to this RCK gating ring and the voltage depolarization modulate the opening of the gate of the BK channel. So structurally speaking, we have cryo-EM structures of both calcium-free and calcium-bound states of the BK, of two types of BK channels. However, what's interesting here is that the intracellular gate, the one that's open right here in MTHK, is always open in both states, in both the calcium-free and the calcium-bound state. So there was a question, and there still is a question, regarding the BK channels, whether these two, calcium-free and calcium-bound state, are actually open states, or whether there is no such thing as a closed state that was determined due to the presence of these voltage sensing domains that seem to mess up conformational states in most channels that I've seen, and structures that I've seen. Or is it that, you know, the gate is, as was proposed in the past, at the selectivity filter, and this intracellular gate doesn't matter so much. So we've decided to go ahead, and this is what I was talking about. So this is the calcium-free BK gate. So you can see that it's wide open at the intracellular bundle crossing. So these are the pore. I'm showing you the pore here of the PK channels. Just two of the opposing subunits shown for clarity in calcium-free and calcium-bound. So in both cases, this is wide open. There is some conformational change here, but it's not sufficient to shut it. So we decided that in order to get to a real closed state with not all the complications of voltage sensing containing proteins, we were going to go to the MTHK channels and that are purely modulated by calcium, and in the absence of calcium, they should yield a closed state. Okay, so we went ahead and proceeded to do cryo-EM structural analysis of the MTHK sample in the absence of calcium. So what we did is we purified MTHK in no calcium. We added EDTA to make sure we get everything, all the calcium out of the way. We incorporate it into nanodiscs, and this is shown here, the nanodisc. Basically, a nanodisc is a little lipid patch that is enclosed by a dimer of apolipoproteins that are shown here in violet, and the lipids are shown in green, right? So these are the bilayers, and in the middle of such a bilayer, you can incorporate any membrane protein. And what we did is we put MTHK in this little nanodiscs, and you can see here the preparation. So it gives a very nice peak. And if you run this on an SDS gel, you can see the full-length MTHK, the channel, the MSP, which is the apolipoprotein, and the RCK domains that are coming, the ones that are produced separately in the cell from the second initiation site. So we have a very nice soluble sample of calcium-free MTHK, which should be in the closed state. So what we did with it is we take a few microliters of this liquid sample, right? And we put it on a grid. We plunge freeze the grid, so we freeze it fast in liquid ethane, and then we put it into an electron microscope. We collect the data, so a bunch of 2D projections of this grid, and then we pick all the particles from the grid. We align them, average them, classify them, and obtain a 3D map of this particles, and then we fit it with a 3D model. So here's the data, and what you see here is one of those micrographs that I told you that this is the raw data, and these little tiny black things here are actually the channels in the nanodiscs. And what you want here is that to have all the possible orientations of this channel on the grid, so that when you take a million of these images, you can obtain all the orientations that would allow you to obtain a proper 3D reconstruction. And we did that, and these are the class averages, all the averages for most of the orientations that we found. You can see that the channel is in all sorts of different orientations. This is the nanodisc, and this is the RCK gating ring, and you can even see here the transmembrane domain. So we could see right away that this is going to go to high resolution, and here's the reconstruction. So this is the final map, a 3.7 angstrom resolution, and this is colored by local resolution. So what I mean by this is that in dark blue, we have very high resolution areas, domains, 2.8 angstroms, and red is around 4 angstroms. So you can see that only the top part and the more external parts of the protein are lower resolution than the ones in the middle. Okay, so here's the structure. This is the density colored by monomer. So each monomer has a different color. This is the model in cartoon representation. Here you can see better what I was talking about before. This is the full-length protein, let's say, in this maroon color here. And this is associating with the cytosolic RCK that is started from the internal start site. And this is the gating ring. This is the pore. And what you can see here that you couldn't see in the structure before is that we actually have the entire Sealinker between the pore domain and the RCK domain resolved in this structure. And this was not resolved in the crystal structure. Another interesting feature here is the fenestration that opens up actually in the closed state in between transmembrane domains, in between adjacent subunits in the middle of the membrane. So this is just a movie for you to see it a little bit better, the structure. And this is going to rotate a little bit, and then it's going to zoom into this fenestration that I think it's actually quite important for the function of this channel. We may not get to that, especially because it's unpublished. And here's the individual subunit with the linker. So this blue thing here is what was not resolved in the crystal structure in the open state. So what you can see is that there are two additional alpha helical turns that extend the TM2 all the way down, and then this Sealinker is a loop that lays flat on the RCK domain. And this is just for those of you that might be interested in individual domains and how they look like. And this is the Sealinker that I've been harping on about. And you can see that there's a possible salt bridge here between this Sealinker that is lying flat. Basically, it's a loop that's interacting with the adjacent RCK domain. So there are interactions that are specific that are formed in the closed state, which are obviously no longer there in the open state. So now the interesting part, right? What does the gate look like? What does the pore look like in the closed state? Is it open like the BK channel, or is it not? And I just gave it away. And this is the pore of the intercellular calcium-free closed MTHK. These are two subunits again, so I'm only showing the two opposing subunits of the tetramer, so you can actually see the pore a little bit better. So this is the pore drawn with the program hole. And when you see red here, that means that water cannot pass through. So here's the selectivity filter part, which is always a narrow area, although we know that that's passing ions. And then here's the intercellular bundle crossing part that is actually formed in this channel, because there is clearly a constriction at this level of this hydrophobics, both I99 and L95 at the bottom. So MTHK, in the absence of calcium, is closed at the bundle crossing. And here's the showing the pore radius in this region. Compare the closed in black with the open in red. So you can see that the open of the crystal structure is quite open. And these two are showing less than 1 angstrom radius, pore radius here. And this is an overlay between the crystal structure in with the calcium bound, which is the open state in blue, and the closed cryo-EM structure in the absence of calcium in red. So again, I would like to highlight this part that was quite interesting. These fenestrations that actually open in this closed state. So this is the closed state, and it's a space-filled model. So you can this is the transmembrane part, and this is that linker region, and this is the fenestration that opens up in the closed state. And in the open state, which is also showing the same space-filled model, it doesn't exist. All right, so this was so we determined the structure of the closed calcium-free MTHK. Now, we thought it was worthwhile to look at calcium-bound MTHK also, even if there is a crystal structure of this protein. And I am very glad that we actually did, because what we found was very interesting. So we didn't find just one conformation of calcium-bound MTHK. We actually found, I think, 9 different conformations, in the sample of MTHK in nanodisk bound with calcium. And I'm showing here just 3 representative ones. And these are differing from each other by having different tilts of this calcium gating ring with respect to the transmembrane region. And this one is the most tilted one that we found, and it's about 20 degrees. This one is 8 degrees tilt, and this one is not tilted at all. And here are the corresponding models. And the highest resolution one is actually the one that's most tilted. And what's interesting about this one too is that it still has the extended TM2 and a Sealinker still resolved. All the other subunits in this particular structure, and the most tilted one, do not have this Sealinker resolved anymore, just like the crystal structure shown, and neither of the other ones have it either. So you can see here that we just put them, we just connected them together with dotted lines. So the first thing that I wanted to attract attention to your attention to in these structures is that the open state looks in the transmembrane region exactly like the crystal structure, meaning that the open state has a large opening at the bundle crossing. So this is the selectivity filter, this is the bundle crossing, and this is an overlay of these structures with the crystal structure, which is in beige. So you can see that they are about the same opening at the bottom. The other thing that I wanted to point out is that we were kind of concerned about the tilts, right? So I was like, what's going on with this RCK domain tilts? Are these physiological at all? Are these induced by the nanodisk? What's going on? Could we reproduce such tilts of the gating ring in a planar membrane? And in order to do that, we did MD simulations, and it's not, when I say we, I mean Toby Allen and Emily Flood from RMIT in Australia did MD simulation of the MTHK closed in a lipid bilayer. And what you see here is, you're going to see a movie. You're going to see this. This is the original structure that we got of the closed channel. This is the bilayer, and this, I'm just showing the head groups here in gray. And here it starts. And the first thing that we saw in this movie is that these Sealinkers that you saw well defined in the structure are actually detaching from the RCK domain and moving towards the head groups of the lipids. And then you also see tilting, right? You see tilting of these gating rings when the RCK domains transiently contact the head groups of the lipid bilayer. So the tilts do exist, they do happen in planar lipid bilayers, not just in nanodiscs. So we think that this is a feature of the protein. So the protein is particularly flexible in the gating ring with respect to the transmembrane region. And also what happens when these Sealinkers contact the head groups, the TM2s are pulled a little bit apart as if they're trying to open the gate. And this is kind of interesting. They didn't manage to open the gate because the hydrophobic tight steric closure still remains at those two hydrophobic amino acids that close the pore at the bottom. But you can see that the amino acid one turn down, the glutamine, actually is pulled apart. And this is a summary of what we saw with the in the MD simulations. We saw the gating ring rocking, lipid interactions with the Sealinker and the gating ring, a bend of the TM2, like I just said, allowing these glutamines to be further apart than in the original structure. And also we saw lipid entry through those fenestrations. And this is just another little movie to show you how much the cues, oops. Let's see if it plays. Yes, how much the cues fall apart during the simulation, not fall apart, but pull apart away from each other during the simulations when the Sealinkers kind of contact the lipid head groups. Okay, so this is shown here. So this is the distance variability, the variability in gate size upon during the simulation, right? So this is time 0. You can see that immediately this Q right here at the bundle crossing becomes pulled away, but the other two, the leucine and isoleucine, which are right here, are doing a little bit of that, but not. They're getting back to being tight at the bundle crossing. But it does give the impression that it must be quite easy to open this gate when the time comes. Okay, so I said to you that the calcium-bound methK structures are open, but they're actually not just open. They are also inactivated. And here's what the pore, the transmembrane region actually looks like in all those calcium-bound MTHK structures. So this is, again, just showing two opposing subunits so that you can see better what's going on in the pore. This is the selectivity filter, and this is the cavity. So what you can see is that the TM1 N-terminus, so this is the very N-terminus of the protein, okay? So the TM1 actually folds inwards and gets into the pore, clearly looking like it's sterically blocking the pore. And this happens in all of the open MTHK structures that we saw. And I should call them calcium-bound methK structures that we solved. And if you actually look at this, at the alignment between MTHK and PK channels, you can see that the N-terminus of MTHK is larger than that of the PK channels. And the PK channels, we know that they do not inactivate without accessory beta subunits. So they need beta subunits to inactivate. So this N-terminus we thought was responsible for this inactivation. And this is just another movie for you to see the structure better, the highest resolution structure of the calcium-bound methK structure, open and inactivated. And this is just zooming in a little bit on that Sealinker part that is still resolved in this open state, and this is the inactivation ball and chain. Okay, so does this inactivation, does this N-terminus look at all like all the other inactivation peptides that have been studied for so long? And here's an alignment of they are all about the same size, 20 amino acids. The methK one that we just solved here is on the top, and this is aligned with the shaker one that I showed you in the beginning, the original one. The inactivation N-terminus for the beta subunit of shaker, which was studied in that McKinnon paper that I referred to earlier on. And these are the two BK channel beta subunits whose N-termini also are responsible for inactivating the BK. And what you see here, and what was known already for quite a while, is that the sequence is not conserved per se in these inactivation peptides. But what is conserved is the character of this peptide. And what I mean by that is that the tip of these peptides are always hydrophobic, and by I highlight the hydrophobic amino acids in green, so you can see them here in green. So all of these have hydrophobic character at the tip, and the base of the peptide is positively charged, and I highlighted the positively charged residues in red. So does this channel inactivate? Right, so if I've shown you an inactivated channel, I need to show you that this channel inactivates, right? So what we did for getting at the function of this channel and seeing whether there is inactivation going on, we did a fluorescence-based ion flux assay using a stop flow spectrofluorimeter. So the way we do this is we take liposomes and we incorporate them with MTHK channels. So those are the purple things you see here on the membrane. And these liposomes encapsulate a fluorophore, it's called ANTS, and this fluorophore is quenched. The fluorescence of this fluorophore is quenched by thallium ions. So it is well known for quite some time that potassium channels and other non-selective cation channels too are actually permeable to thallium. So when channels are open, you can imagine thallium going in and quenching the fluorescence. So that's the basis of this assay. So what I'm showing here schematically is that on one arm of the injector, I'm loading, for instance, with this ANTS loaded liposomes with methK on the side. In the beginning, they are in the absence of calcium, so all the channels are closed. And then through the other arm, you give thallium which is the quencher, in addition to whatever activates the channel, which is the case in this case is calcium, right? So let's say we have calcium and thallium here, and here are the liposomes, and you mix them quickly, and when you give them calcium, channels activate, and the thallium goes through. And what I've just been showing you is what I've just been telling you is shown here. So in low activity, like when the channels are not very open, you see slow quenching of the fluorescence. But when the channels are very open, you see high quenches, fast quenching of the fluorescence. So what we do, just quickly to tell you how we analyze the data, we take the first 100 milliseconds of this fluorescent quenching curves, and we fit them with stretch exponentials, and then we take the rate at 2 milliseconds. Okay, and so everything that you're going to see is plotting the rate at 2 milliseconds from such curves. So, does the channel inactivate? So we, in this case, we used very high calcium concentrations to activate the channels, which activate instantaneously within the at that time of the instrument. And then you can see that over a period of about, with a time constant of about 1.5 seconds, these channels actually inactivate, right? So you see less and less thallium flux rate. Now, if this segment, if this first 20 amino acids are responsible for inactivation, if you cut them off, you should remove inactivation. And this is exactly what happens. We made a construct without those first 18 amino acids, and this particular construct is no longer inactivated, as you see here in red. So we also did the structures of these non-inactivating MTHK, and what we found is that we have, in the presence of calcium, what we found is open channels, but without the inactivation peptide. So we also did, meaning Emily and Toby, did MD simulation to figure out how favorable it is for this peptide to bind in the pore. And basically, they found that it takes quite a lot of energy to pull the peptide out of the open pore. So they started with the structure, and then they tried to pull it out. And this is steered MD simulation. And what they found that it takes about 18 kcals per mole to pull the peptide out. And analyzing the interactions in this particular simulations, we find that the base, which is the positive part of the peptide, interacts with this glutamates at the intercellular lower base of the channel. There are two rings of glutamates, and the tip, which is hydrophobic, interacts with the hydrophobic cavity. So just quickly, so I believe I'm out of time. Just quickly to summarize, so this is the summary slide. What we've determined here is the key conformational states in the calcium, in the MTHK calcium gating cycle. So in the absence of calcium, the channel is closed. It's closed at the bottom. The RCK gating ring is in a apo resting state, and as I said, the channel is closed here. We have no idea where the N-termini, where the ball peptides are when they're not in the pore, and the Sealinker actually and the extended helix are actually present in the closed state. And this is the structure that we obtained in the absence of calcium. When calcium is applied, there's a conformational change that occurs in the RCK gating ring, which pulls on the TM2s in some manner, and it leads to disordered Sealinker and to the unfolding of the bottom two helix turns of the TM2. Again, the N-terminus is not visible because it's not in the pore, and the channel is open at the bottom. And slowly, this channel inactivates by this ball eventually making its way into the pore, and we have this structure for this here. And this was published early last year in the middle of the first wave of the coronavirus pandemic. So I would like to thank the people who did the work, the people in my lab without whom this wouldn't have been possible. So Chen and Yan did the cryo-EM structures of MTHK with and without calcium. Yan is no longer in the lab. B did the functional assays with both the single channel recordings that I didn't show you and the stop flow recordings for the inactivating and non-inactivating channels. Xiaolong, Elizabeth, and Philip are working on different projects. NIH, DFG, and American Heart for funding, the New York Structural Biology Center and the cryo-EM core facility at UMass for allowing us to collect the data, and Toby and Emily for the MD simulations at ARMIT in Australia, and all my other collaborators whose data I didn't show you here. And I'm going to leave you with a picture of my lab pre-pandemic, and I would like to thank you all for your attention. Thank you.
Thanks, Crina, for this very nice talk. So we have a few questions. So the first is from Mark Sanson, who asks you, is there any density showing whether lipids could block the fenestrations?
Yes, so we have some data that shows that, so basically we have a higher resolution structure of the calcium-free MTHK that is not published yet. And that one shows that there are lipids that make their way into the fenestration. They have one of the tails inside the fenestration. The other tail is kind of wiggling around. It doesn't lock it per se because we give it calcium and the channel opens. So, but yes, you could imagine lipid modulation given the interaction with the fenestration.
Good. So Hannah Moroni was asking you, if you have the peptide, do you reestablish inactivation in a mutant that lacks it?
Yes, yeah, we have those. It's a very good question. So we have those experiments. Again, they're not published. So we have synthetic peptides that we made of the 19. We made 19 and 13 amino acid peptides that mimic the N-terminus, and they block. So 10 micromolar of those peptides, of one of those peptides, the other ones are not very soluble. The 19 mer. They block very well with the time constant of the physiological inactivation. And if you make one change, you change one of the hydrophobic tip into a glutamate, you lose block at 10 micromolar. We haven't done higher concentrations, but it's interesting that it's very specific to this hydrophobic tip positive charge bottom.
I see. Sergei Sukharov asks, what is the experimental time of recovery from inactivation?
I'm sorry, can you say it again?
Yeah, what is the experimental time of recovery from inactivation is a question by Sergei Sukharov.
We don't really measure the recovery from inactivation. We only measure what I've shown you, which is the equilibration between open and inactivated.
You see. Enzo Carnevale is asking if does the tilt show any dependence on the lipid composition of the membrane? And is the channel more constrained in a thick raft-like membrane? In
a what membrane?
In a thick raft-like membrane.
Oh, in a thicker membrane. Yeah, so we've been doing this, very good questions. So we've been doing these experiments because we've been trying to also figure out there is very strong dependence of the inactivation time constant on the thickness of the bilayer. So basically in C14 lipids, it inactivates in milliseconds, and in C22 lipids, thick lipids, it inactivates in minutes. So it's a very strong dependence, and we don't quite know what that is due to. We did do the structures in C22 lipids, and I think that what we've seen, it's not done yet, but we do see fewer tilt conformations. Yeah, I'm not sure that I can draw that conclusion that that there is a tilt dependence. I think I need a little bit more data for that, but it's a very good question.
I see. There is another question from Anna Moroni.
Mm-hmm.
Why the peptide was not visible in the crystal? If was it there, or did they remove to favor crystallization?
They didn't remove it, or at least they didn't mean to remove it. They could have mistakenly, but they didn't. I don't know why it's not showing in the crystal, but my suspicion is that, as I try to evoke, is that the crystal is that of a mutant. We don't really have. I believe that there is no real physiological open state that it's captured by that mutant. So it is quite possible that the tilting of the RCK gating ring and the allowing the protein to move may actually allow the entry of the peptide in. But this is all speculation. Another thing is that the protein was crystallized in a way that there's very little space in between the RCK domain and the pore domain, so there may not be enough space for the peptide to sneak itself in. So there are possibilities, but I don't know which one is correct.
So I have last question from Blasco Morozzo. What about the fourfold multiplicity of the plug peptide, if this does play a role?
Well, right. So if you would have, right, so if any of the peptides is allowed to come in and bind, then if you have 4 of them, you will get 4 times faster inactivation, right? If you have just 1, it will take 4 times longer. So physiologically, it is important because it modulates the time constant of inactivation, if that was the question.
I see. Okay, I think that we can. I don't have any other question here, so we thanks Crina once again.
Thank you.